Hi, everyone who joined. It's my great pleasure to see many people joining today and to introduce to you Dr. Erin Baggs. Woohoo! I'll give you a few slides. I've known Erin for over 5 years now. This is the pictures from her for one of the first months she decided to come into the lab. She decided that she wanted to be not only a computational biologist, which she excels at, but also a molecular biologist. And she started here. I think like the sweater she was wearing that day really well summarizes Erin's personality of continuous growth and being very cheerful about it. So I remember I'm glad I took pictures of that back then. And that was United Kingdom. So Erin started with me as a first PhD. She hedged her bets on me being an okay PI. She was a direct admit to the lab. And moreover, it was within her first year that I told her, you know, Erin, I'm going for an interview at UC Berkeley. How do you feel about it? And Erin was one of the few people who decided to make the jump between UK and UC Berkeley. And, you know, I'll show you all of the scientific accomplishments that Erin has done. And all of you know that she worked very hard. She's so hard worker. But behind the scenes, she and Andrew were also helping to establish pretty much like a brand new reopening lab and moving all of the seed stocks and getting us up and running. Now, at the same time, Erin was taking classes at Berkeley, and she passed her Berkeley quals in under 2 hours. So typically our qualification exams take 3 hours. They're long, they can be brutal. The committee basically kicked Erin out halfway through, said, you know, we're done, you're qualified. This is Erin prepping for the quals a few days earlier. She mentored many people. She mentored Ruby in the UK and 3 undergraduates at Berkeley during her time here. She trained them and they've become really good scientists and they've all valued their time in the lab. She also mentored rotation students and taught and won the Outstanding Graduate Student Instructor Award. So that tells you a lot about Erin being really a good mentor as well and investing her time in it. And finally, for those of you who are really nitty-gritty scientists here, this is not a list of my favorite journals. This is all the journals that Erin's research has been published in already. So she's an accomplished multidisciplinary researcher. And from what she'll tell you today, you'll see that 2 other papers and 2 other stories are actually yet to be published. And if you're watching this recording that we may or may not put online, or at least my introduction to it, and you're an editor, your journal name can be here. All right, that's it. I'll give it to Erin now. And thank you all who joined during my introduction.
Thank you very much, Ksenia. That was really kind. Thank you. So now hopefully you guys can see my screen. And I'm going to, first of all, thank you for taking the time for coming to my finishing talk. It may be a little different to a normal talk because our audience is very varied, from friends and family to other people that I do science with. And so for this fact, there will be some generalizations for the sake of time or to try and make it kind of approachable. But what I plan to talk about today is lessons from loss, plant immune signaling and pathogen interactions on land and in water. So I thought the most apt way of taking you on a journey through my research was using the tube map. So many of you know I grew up on the last stop of the district line, the tube in the UK. So this is very familiar to me and is a nice homing sign. So we'll go through the talk in zones. And at the end of each zone, there'll be kind of an intermission video to give us a little break from the science. And to start with, we'll have an introduction on how I got here today and some background. And then we'll go into research chapters of can immune pathways be lost? And then what happens to remaining genes when they are lost? And then kind of shifting gears a little bit to look at the development of a simpler plant disease model before making it more complicated again by adding the environment back in. And then finally, really the most important part to me is the acknowledgements at the end. So first of all, how did I get here? And as Ksenia mentioned, it wasn't the most straightforward of journeys. So in 2016, I started as a PhD candidate at the Earlham Institute and TSL in the UK. And I did my first sort of qualifying exam, my probation exam. I took a number of courses. And then it became clear that the Krasileva Lab was relocating to UC Berkeley, which was an amazing opportunity. And me, Andrew, Ksenia, and thousands of test tubes and seed packets traveled over to the US. And then I had to restart some things. So I took more graduate modules, did another qualifying exam, and then got the chance to go to some conferences in person and teach before the onset of COVID. And then after that, I did more teaching, more research until in December 2021, I submitted my thesis. And then the last stop on my journey really is today, giving this thesis talk. So I study plant immunity, but why do that? Well, 1 in 9 people in the world suffer from hunger. Plants produce over 80% of the food we eat. And they also provide nutrients from our diet. And beyond this, they also can be used as factories to produce drugs and vaccines. They can help us maintain a healthy environment. And however, although we depend on them for all these different things, we lose a lot of crops every year to plant diseases, 220 billion roughly. And our problem of plant shortage is only going to get worse because the population is expected to grow and higher living standards demand even higher crop production. So we're left in a situation where we need more plants and we don't want to lose as much anymore. So it's really important that we can make our plants healthier and more resistant to disease. So that's why I studied plant immunity. But how do I study it? Well, there's kind of 3 pillars of logic behind my research. And the first is that plants are diverse. And we typically only study a few plant species, normally Arabidopsis or rice or tobacco. And so we're just focusing on these few species. And often they have problems and we don't understand why they do things in certain ways. And it's only when we look at the diversity of all the other plants that we may be able to find solutions for these problems. However, comparing a lot of plants is very difficult. It would create a lot of need for growth space. So how do I do this? Instead, I rely on comparing the genomes. So we can think of genomes as books, because genomes are also a collection of letters, A, C, T, and G. And they're basically all the DNA you need to create a living organism. And genomes are typically divided into chromosomes. And within those chromosomes, you have thousands of genes. And genes can be thought of like recipes. They give us the instructions of how to make things that are important for the cell. And when we want to use one of these recipes in biology, we go through the process of transcription. And we can think of this as photocopying the recipe. And it gives us something to go off so that we can produce our end product, which is normally our proteins. These are kind of the essential molecules in the cell carrying out those molecular machinery. And so I compare genomes, and specifically I'm interested in pathways. And so pathway complexity varies between species, and pathways are inherently complex. So in this diagram at the bottom here, what you see is there's a surface receptor, and there's a number of different pathways before we get to the output. And the fact that these pathways overlap makes it difficult to understand any one pathway. So I'm typically interested in species that have lost a particular pathway, because this offers us 2 avenues for understanding one without a particular pathway, we may be able to understand what remains better. And also, everything that is part of that pathway is likely to be lost. So if in our understanding, we're kind of missing a few steps, when we study species without that pathway, it can give us hints to other members of the pathway. And so they're kind of the three: logic, diversity, genomes, and pathways. But how do plants defend themselves from disease? Well, if we start at the level of a plant cell, if we think of the plant cell, there's a lot going on in there, and I didn't want to draw it all out, so I'm just putting chaos. So bear in mind, there's a lot going on in the background that I'm not really referring to here. However, when a plant cell encounters a pathogen, in this case, a bacteria, there are receptors in the cell known as NLRs. So these are proteins, and they're able to detect the presence of that bacteria and trigger an immune response. And this immune response can be dependent on other proteins, such in the case of this NLR here that requires EDS1 in order to create an immune response, or it can use other pathways that are part of that chaos. So importantly, there are different types of NLRs, CNLs, TNLs, RNLs, for instance. And these NLRs typically work in different pathways. So we know that our TNLs typically signal through this EDS1 protein and RNLs in order to cause a disease response. So this will be something that we hark back to throughout the talk. And so this is the end of our first zone. So we have a little intermission. And this is my dog back home, giving her opinion on her Boris Johnson toy. So she's not the biggest fan, like most of us right now. So now we're going to move on to our first research chapter about, can immune pathways be lost? And before I dive into the science here, I want to kind of go over some concepts that might be useful to understand. And the first is that taxonomy. So how we name a species often has to do with its relationship to other species. And so species is normally one of the more stringent names that we give. And then going up, then there is more. So in the family Enterobacteriaceae, there'll be a whole load of different species, and one of those species is E. coli. So this is how taxonomy works, the naming of systems. And we can also name plants in a similar way. So eudicots include a number of plant species you're probably familiar with, such as tomato, pepper, tobacco. And these diverged quite a while ago from monocots, so we give them different names. And monocots include species like banana, oats, rice, wheat. So we'll look at these two different groups throughout the talk. And you can see that the relationship between these species is shown here through a phylogeny. And when you read the phylogeny, those branches that converge closest to the right are more closely related. So here eudicot and monocot are more closely related than monocot to gymnosperms, which include kind of conifers. So we can use this to also understand the relation of different organisms. So I'm interested in this question of, can we lose an immune pathway? And if you lost an immune pathway, would you still be able to survive, or would basically the pathogens take over? And so my first step to looking at this was comparative genomics of a number of plant species with the names on the left here. And then I looked at the number of those sensor genes, those NLRs, on the right. And what we saw is that there is a really variable number of NLRs in different species. These are particularly low NLR species. And we already knew that some TNLs had been lost at the base of the monocots, but we were interested in if beyond that there had been loss of particular NLRs. And so some species, such as resurrection grass here, that has really few NLRs, has still retained the different types. So they have RNLs and CNLs, but others, such as these species in blue, had both lost RNLs, so had only retained a CNL type of NLR. So given that they lose specific types of NLRs, we were interested in whether they had lost specific pathways. And so as these species highlighted in red don't have TNLs or RNLs, we were interested in if they'd lost this whole EDS1 pathway. And so to show this, we use a dot plot here where a black filled in dot shows the presence of an immune gene that's part of a signaling pathway, and the gray is the absence. And so what we see is that indeed in these two groups of species, we have lost EDS1 PAD4. And we know these losses must be convergent. So it didn't happen one time, it's happened twice, once in this lineage and once in the other lineage, because they're in different groups on the tree. So the greens are monocots and the bottom ones are eudicots. So now we know we can lose a pathway, we wanted to know more about that lost pathway. So to do this, I tried to find other members of the EDS1 immune signaling pathway. So I took all the proteins from diverse species and asked for only those protein families that were present in all species that do have EDS1 and absent from all species that don't have EDS1. So that includes like our duckweed. And then when we did this, we got to a list of 52 proteins. But I had also just found out that asparagus had lost EDS1. So we further filtered our list through asparagus, and this left us with just 16 proteins that are present in all species with EDS1 and absent in all species without EDS1. And these proteins are down the bottom of the slide here. And many of them were already known to have a role in immunity, but SDR4 in particular, shown here, was not. And so when we looked at RNA-seq data to see if this gene has more photocopies, for instance, when we add a pathogen, we see that our candidate SDR4 here has a very similar pattern to the known immune genes. So this makes SDR4 a really strong candidate as having a role in immune response. And so in this section, I have shown you that plants can indeed lose immune pathways, in this case, the EDS1 pathway. And we use correlation of the presence and absence to predict novel genes involved in that same pathway. So we now get to our next intermission, where this is the puppy that we got during COVID, Luca. So he thinks he's a little singer. I can't take credit for the saxophone player, that's not me. But now we'll move on to the next section of do lost and conserved pathways work in the same way? So earlier I told you that we know a number of species that have lost these TNLs that function through EDS1. But even in these species that do not have TNLs, we noticed they had a very similar protein called a TMP. And so previously we'd kind of grouped these TNLs and TMPs together, thinking they were the same thing. However, we see TMPs present even in species that do not have EDS1. So this doesn't really make sense. How do they signal? And another important fact is that TNLs, their disease protection response pathway is often cell death. So they often cause the cell to die, and this often releases chemicals and different compounds that can prevent the pathogen from spreading any further. So we wanted to see about if TMPs are highly conserved across different plants and if they show similar genomic characteristics as TNLs. So I've highlighted in yellow here the TNLs and TMPs, and you can see that the TNLs, the size of the dot refers to the number of the copies of a TNL type gene in the genome. And you can see that in some species there's none, in other species there's up to 100, so there's really wide variation. Whereas our TMPs are very consistent. They're the same size dot throughout. They're in all seed plant bearing plant species. So it appears there's different genomic signatures between these TNLs and TMPs. So next we wanted to see if they would have similar function. And to do this, we engineered some bacteria that is able to produce these proteins. And we injected that into the tobacco leaf. And when we do this with TNLs, we often also add a trigger protein, and then they cause cell death. So this is this black gray kind of lesion here, whereas in the control, we just see a beautiful green leaf. And so we tested a number of our TMPs to see if any of those cause cell death. Most didn't, but this could be because we're missing a trigger. However, one, even on its own, was able to cause cell death. That was this maize TNP1. Okay, so cool. They're different from TNLs, but they still share this property of being able to induce cell death. Next, we were interested in if that cell death was dependent on EDS1, as is TNLs. So we were able to use a tobacco plant with RNAi to suppress the production of EDS1. And this means that you can't photocopy that EDS1 gene and you can't produce the protein. So there's basically no EDS1 protein in those cells. And what we see with our TNL is that we lose the cell death response. However, with our maize TMP, we are able to retain the cell death. So this means that our TMPs are not really the same as TNLs and instead signal through a different pathway in order to mediate cell death. So in this section, I've shown you a genomic example of the uncoupled of presence and absence of a gene in the same pathway and how we were able to use this to predict and then show that TMP genes can induce cell death independently of EDS1. And so for our next intermission here, we have my UK COVID puppy that's with my family back home. And this is Breezy. And she's decided to take our older dog Floss for a walk.
Oh, good girl.
As you can see, Floss isn't too sure about it and decides that she'll take herself for a walk. But you can see Breezy is also very studious like me. So can we develop a simpler disease model? So after all the work with tobacco, I was kind of tired of these big plants taking up so much room and taking a long time to grow. So we wanted to see if we could make it any easier for ourselves. And one of the important tools that we use when studying plants' response to pathogens is RNA-seq. And this is where we're able to read out how much of these RNA photocopies are made. And this is important because this normally relates to the amount of protein which are often created in response to a stimulus and can allow the plant to adapt. And so we know that immunity is very complicated, and we wanted to see if we were be able to make it simpler and easier for ourselves. And for this, I chose duckweeds because I knew from earlier that Spirodella, at least, didn't have EDS1 and RNLs, so it had lost one of the pathways. They also have a reduced body plan of a fused leaf and stem. So if you hear me saying a frond, it basically just means the leaf on Spirodella or other duckweeds. And they're tiny. So they're as small as 1 millimeter, which means in the space that we would be able to house, say, 50 Arabidopsis, we can have thousands of individual duckweeds. And moreover, they can double in as little as 39 hours. So if I accidentally kill some of my plants, I don't have to wait that long before I can get a new lot. So duckweeds have lost several immunity genes. But I wanted to map this out a bit more because at the time I only knew about Spirodella, so this specific duckweed. And so in this diagram, what you can see is a plant cell and then a number of immune components. You don't need to worry what these immune components are, but the takeaway message is that there are kind of less of these known immune components in duckweeds, and that even within duckweeds, there's further reductionism, with Wolffia having even less than Spirodella, for instance. And we can map this out more quantitatively, where we have immune genes along the top. And then in the columns, there's a bubble where the size of that bubble represents the number of those immune genes. So in the case of RLPs, we have 213 in rice, but we have much fewer in our duckweeds. And the general takeaway is that duckweeds have less of a number of these immune gene families, or they just don't have the proteins at all. And there is one contrast to that we found, which are these antimicrobial proteins, which are expanded in a number of duckweeds, but this specific type is not even present in Arabidopsis or rice. So if we want to study disease on duckweeds, we first need to see that duckweeds can get diseased. And so here I have a panel of my different duckweed species, all very healthy, nice and bright green. They've filled out the wells. And then if I inoculate them with a pathogen, in this case, Pseudomonas syringae pathovar tomato DC3000. So I'm going to come back to this pathogen later, and I'm just going to refer to it as Pseudomonas just for simplicity. But this is its full name in case you're interested. And you can see this causes black dots, clumping of fronds, and it's not able to grow as well. And we tested a whole range of pathogens, and in general, we see a lot of susceptibility, and we do see variation across the duckweed species in their response. So next, we were interested in how the Pseudomonas causes disease and whether it would be the same as in other plants. So if we look at the surface of a plant cell, there's these little O kind of shaped holes, and these are stomata. This is where air gets into the, air can be exchanged for the plant, and also often where Pseudomonas enters. And then below this top level, we have subsequent several layers of cells, which are performing different functions in the plant. So we did some microscopy to look at this in duckweed, and in gray is the plant cells, in green is chloroplast, and then in pink are Pseudomonas. And so we're going from the top of the leaf to the inside. And what we see that there is indeed duckweed within the leaf. So it's not just sitting on the surface there, it is indeed inside the plant. So now we know that Pseudomonas gets inside, we're interested in how it is able to cause disease. And one of the main ways that we know Pseudomonas causes disease is through its type 3 secretion system. So you can think of this as like a syringe that is able to inject these effectors, which are often able to manipulate the plant cell. There are, however, Pseudomonas which have had a gene deleted, so are no longer able to make this syringe. And this kind of traps these effectors in the cell so they can no longer modulate the plant. So as Pseudomonas isn't thought to be adapted to duckweed, we wondered whether these effectors really were having any sort of role in disease progression. So when we see wild type infection, we see these black lesions as early as 3 days. And very quickly, the duckweed looks unhealthy. However, in the HARC-C mutant, which isn't able to produce this syringe or deploy its effectors, we don't see these symptoms advance as quickly, although if we wait maybe 4 weeks, we will eventually see the discoloration. So we also wanted to look not just at the visual symptoms, but how much of the bacteria was growing, and we're able to do this through growth curves, where at 3 and 5 days, we crushed up duckweed tissue that was inoculated with these bacteria and asked how much of the bacteria was there. And what we see is that there is far fewer numbers of these bacteria which have lost their syringe than those that have it. So indeed, these effectors seem to be playing a role in disease progression in duckweed and the number of colonies. So now we have this Pseudomonas disease system we're using. We wanted to see how the duckweed itself is responding to this infection. And to do this, we looked at RNA-seq. So we are comparing between duckweed, which we've treated with a pathogen, to duckweed, which we haven't treated with the pathogen. And we're seeing how many copies of RNA of each gene are in one versus the other. And so if we see more RNA of a particular gene, the gene's colored red when treated with pathogen. And if we see less, then it's colored blue. And there's a number of genes that we expected to see as broadly red because of work in other species. But this didn't appear to be the case in duckweed. There was a lot of variation in which genes were up and down regulated, and it wasn't necessarily those that we expected. So next, we really wanted to hone in on what is conserved and upregulated across duckweeds and also other plant species such as Arabidopsis that have more of those immune pathways. And so we looked at this intersection of these 28 genes that have been upregulated in our 2 species of duckweed after treatment with Pseudomonas. And we saw that 4 of these genes are also upregulated in Arabidopsis when treated with Pseudomonas. So there are some highly conserved upregulated genes across both monocots and eudicots. However, we were still kind of interested if duckweed's not really responding in the normal way we expect, then how is it responding? And so I went back to these antimicrobial proteins that we saw were expanded in duckweed. And sure enough, here we see much more of these dark red, high upregulation in comparison to other genes that in other species we would normally expect to see upregulated. So it appears that these antimicrobial proteins are definitely one way that duckweed tries to respond when it's infected with a pathogen. So in this section, I've shown you that duckweeds do not have some of the typical immune components that we expect to see in plants, and that we were able to develop a bacterial plant system in duckweed where we could identify the differential expression of AMPs in response to bacteria. And so here we have Luca, a little bit older now, having fun in the tar host larvae.
Ready? Ready? Way to go, way to go. Got it. You got it, Eva.
So now we're going to move on to our final research section of, does the environment affect our simple system? And one of the things we're considering when we're talking about the environment is the bacterial microbiome. So a microbiome is the microorganisms, including bacteria, that are present in a certain environment. And each individual bacteria has a sequence known as the 16S, which allows us to tell kind of which group, which species or genus that bacteria belongs to. So we can think of it as similar to a fingerprint. And we can utilize this when we're trying to understand what bacteria are present in a certain environment. And we're interested in what bacteria are present in an environment because bacteria can help plants in a number of ways. They can help them survive drought stress, they can mobilize nutrients for them, but they can also protect them from pathogenic bacteria. They can do this in 2 ways. One is indirect, where they stimulate the plant to protect itself more from those pathogens, or it can be direct interaction between bacteria in the environment and a pathogen through production of antimicrobials, for instance. So why were we interested in duckweed and the environment? Well, it was because in the lab, we saw that duckweed was really highly susceptible to these different pathogens, which duckweed is likely coming in contact with in nature because it exists on all continents except Antarctica. And it is an invasive species, so it's commonly found all over the place. And so there seemed to be a contradiction between the prevalence of duckweed, but also its high susceptibility to disease in the lab. So we wanted to see if the environment was modulating this. And along with a PhD student, Grace Stark, we went to UC Botanical Gardens and sampled some natural duckweed populations to see what we could find. And so we took these duckweed from the ponds, we vigorously shook them in a solution, and then vacuum filtrated that solution to recover just the remaining bacteria, which we could identify the genus from the 16S sequence and understand a bit more about the bacteria that duckweed was in contact with. And so when we looked at the bacterial families, we noticed the presence of a number of bacterial families that are commonly associated with terrestrial plants. So it seems like there's a similar community structure. But if we just zoom in on 2 groups here, we see in the first one, the Rhizobiaceae, that there's a consistent amount of bacteria across the 3 sites. So site 404, site 405, and site 923 all have similar amounts of this family of bacteria. You can tell this because the dot is a similar size and the same color. Whereas Pseudomonaceae varies between the different sites. with the purple showing that there's a high amount in 923, but relative to other families. In contrast, at site 405, we see a reduction. And we were able to show that between sites, we could separate them based on their bacterial profiles. And we were happy to see we could separate site 404 and site 405, despite the fact that they're geographically very close together. So next, I decided to dig a bit further and see if we saw any genera that are associated with pathogenic lifestyles in our duckweed microbiome. And sure enough, we do. Some, such as Xanthomonas, we see present in low number and in some ponds, but not others. But Pseudomonas, for instance, is in all the sites and has less in site 405, as we showed with the Pseudomonaceae in general. And despite these genera being present, we can't necessarily say they're pathogenic, but it's still good context. And so next we wanted to see if the pond environment can protect duckweed. So if we take our normal duckweed and inoculate with our Pseudomonas pathogen, we see we get these black lesions and stunted growth. However, if we just put our duckweed for 24 hours before in pond filtrate and then add in our pathogen, we no longer get disease. So it did seem that the pond water was protecting the duckweed. However, if we filtered the pond water a little bit further, we lost that protective effect. And so given this difference between the two filters, we were interested if there might be some small bacteria present that were facilitating the protection. So sure enough, I was able to isolate a number of bacterial strains from the pond water. And the number at the front dictates the site. So this is from 404 and this is from 405. And then the letters are just to distinguish different strains. So if we treat our lab duckweed with bacteria from sites 404, we see that these are largely pathogenic and they cause these disease symptoms. We don't see that with our strains from site 405. However, if we then co-inoculate with our pathogen, we see disease as we would expect in our buffer and with our site 404. But then interestingly, some of our site 405 strains, 5BF, 5BT, and 5C were able to protect duckweed from severe infection. And so we decided to investigate 5BF and 5BT further, as these were able to survive in the lab very well. And so we next looked to see whether duckweed from that natural environment, so some of the duckweed we'd taken from the pond, if this was able to survive infection. So we had to use a different pathogen this time. But again, when we inoculate our biocontrol strains, so 5BF and 5BT, with the pathogen, we see it clearly protects from disease symptoms. So this took us to the point of, well, is this a direct biocontrol or an indirect biocontrol? So is it that the pathogens are stimulating duckweed itself to battle off, if the biocontrols are stimulating duckweed itself to battle off the pathogen, or is it that the biocontrols are directly inhibiting the pathogen, even in the absence of duckweed? So to do this, we took duckweed out of the picture and we did interaction assays between the pathogen and the biocontrol strains. And we saw this interesting phenotype of the growth of the biocontrol along the pathogen streak. And we don't see this when we cross the two biocontrols. Instead, there seems to be a zone of inhibition. So next we went to liquid culture assays. And in this plot, I would just focus on the blue. And these blue bars show that Pseudomonas on its own has quite a high number of colonies. And if we grow it together with E. coli, we can recover a reasonably high number of colonies. However, when we grow 5BF or 5BT together with Pseudomonas, the amount of Pseudomonas we can recover, which is shown in blue, rapidly drops. So this shows that there seems to be some direct biocontrol happening. And there's a number of ways this could be occurring, but to try and zoom in a bit further, I assembled the genomes of our two strains, 5BF and 5BT, and looked for the presence of genes that would encode these direct biocontrol factors. And sure enough, we see many different symptoms in the two strains. Some they share, but they could also be using different means to protect against our pathogen. And so in this section, I've shown you that duckweeds are very susceptible to disease in lab, yet they are prolific in nature, and that maybe this is due to the protection from the microbiome. And so this is our final intermission. And we have football team UK versus team USA. So this is Flossie, who can even header the ball these days. So her play is pretty awesome. And then we have Luca trying to play American football, passing to Evan, the quarterback. Still needs a little work, but he tries. And so with that, I've now finished the research side of my talk, and we can move on to the acknowledgements. So in terms of scientific acknowledgements, sorry, that went wrong. In terms of scientific acknowledgements, I would like to thank my project co-workers and mentors, including Ksenia Krasileva, Christian Schudomer, and Wilfred Hertie. So these people all followed and continued to support me when I was at EI or at TSL, also Andrew and Yanina as well. And then also I was able to work with a number of great colleagues, such as Grace, who we did the duckweed project together, and Mark, who also really helped me keep going with duckweed. And before that, Ruby, who was like the first undergrad grad student that I got to work with, which was awesome. And so in general, just the thank you to the Krasileva Lab members, and also to my thesis committees. So Russell, Ben, Sarah, Wilfred, Christian, Jonathan, and Kok van Oosterhout, as well as support services that were really crucial, from high performance computing to greenhouse to microscopy. So people were all very patient with me. So I tend to be not a specialist in any of these areas. I was also able to work in a number of great teams, some of which I didn't have time to mention, but these people really helped me. And we also couldn't have done the work without the funding and the support of the different institutes that I've had the opportunity to work at. Also, I'm really thankful for UC Berkeley Botanical Gardens and the Indigenous people of the Lone Tribe, which allowed me to do this research in Berkeley. And so now I have more of my pictorial acknowledgements, but I don't know if we want to do questions first. I can just go on to the pictorial acknowledgements. So first, this is the lab when I joined. So it was Christian, who's been an awesome mentor to me, Ksenia, who's also really supported me, allowing me to come over to the US and really do some crazy projects that I don't think anyone else would have, like my duckweed work. And then me and Andrew joined at the same time. And Andrew was kind of my partner in crime in the first part of the move, when we were dealing with difficulties of assembling tables and hosting a lab warming party, which I never thought would be something I would do. But the lab has changed over the years. We've gained a number of awesome new members that have continued to really create a great environment. And I also want to thank my friends back home who both supported me when I was there and also whilst I've been here. So I met a number of great people during my PhD in the UK, and they've really helped me throughout the whole process. And also my friends back home who really supported me when I decided to move, and threw me an awesome leaving party. And then I also had an amazing opportunity of making more friends out here. I was lucky enough to be part of an absolutely awesome cohort who have really took me in and have been really supportive. Unfortunately, most of our photos are 2 years ago now, but I think that's quite understandable. I also made a number of friends outside of science here that have really helped to keep me grounded and going. And then I also want a huge thanks to my family, my extended family. This is my cousins who have always been hugely supportive of what I do, even going to play mad volleyball games in the UK just after Christmas. And also thanks to Evan's family, who kind of looked after me over here when we've had a lot of family occasions that it's no longer easy to travel for. And then finally, my family, who have been absolutely amazing and super supportive. My parents all dressed in cow stuff, and they also put a cow bandana on my dog, who's so fluffy you can barely see it. But my parents have always been hugely supportive. My mom is one of the most empathetic people you can meet, and my dad is one of the most optimistic. So this is a great support network that I need. And then I also have my 2 great sisters that have always been huge cheerleaders of everything that I've done and really encouraged me to keep going. And then we also have Evan and Luca, who have been my little support system here, keeping me happy every day. So with that, I'm happy to take any questions anyone may have, and thank you so much for joining my talk today. I really appreciate it.
Great job, Erin.
Thank you.
Truly excellent.
Thank you, Sarah.
Erin, do you want me to record the questions or stop here?
You can
your-
stop if you want. Yeah, I'm
No,
sure.
no, but for you, like when you'll have it for years.
Maybe stop, because I don't want anyone to feel
Okay.
pressured that they're